Good morning, everyone. Respected sir. Firstly, I thank my professor, Dr. Muralidhara Rao sir, Department of Biotechnology, for giving this opportunity to present on this topic. And the title of the topic is Post-translational modifications. Today's agenda on post-translational modifications include introduction to the translation, protein modifications, and different types of post-translational modifications, the significance of PTMs, defects, and summary. Introduction. Before entering into the post-translational modification, we should know what is translation. Translation is the process of translating the sequence of messenger RNA, in short it is called as mRNA, to a sequence of amino acids during protein synthesis. And this translation involves three major steps: initiation, elongation, and termination. As we all know that DNA is transcribed into mRNA through a process called as transcription, and then this mRNA is translated into the protein by a process called as translation. In the process of translation, messenger RNA works together with the transfer RNA, it is tRNA, and the ribosome for the process of protein synthesis. Protein modifications. Gene expression is a complex process which is tightly regulated in order to produce an accurate and fully functional protein. So there are three types of translational modifications. There are pre-translational modifications and co-translational modifications, and the last one includes post-translational modifications. The pre-translational modifications are also called as post-transcriptional modifications, and the co-translational modifications are the protein modifications that takes place during the translation process. Therefore, these modifications happen during the protein synthesis. The post-translational modifications are covalent or enzymatic modifications that happens after the translational process. And these modifications can add additional variations and diversity to the type of proteins our body can produce. And this has been termed as proteome. Just like genome, which represents all of the gene that can be expressed by an organism, the proteome is all of the protein that can be expressed. And the really interesting thing is that though our genome is incredibly expansive and has thousands and thousands of different genes, the proteome is actually greater. And the reason for this is that we can have variable splicing of different mRNA transcripts that can influence different proteins we can produce. But also when we produce these proteins, there are ways to control their expression and functions by providing additional modifications to these proteins. And thus the number of proteins that the human body is capable of producing greatly outnumbers the protein we have. And these modifications, which we'll get into in more detail in a moment, can involve things like methylation, that means the addition of a methyl group, and acylation and adding of an acetyl group, and ubiquitination, which is important in degradation of proteins, and many other different modifications. And now let us see the differences between the co-translational modifications and the post-translational modifications. As we have discussed earlier, co-translational modifications occurs during the translation process, and post-translational modifications occurs after the translation process. And the location at which co-translation modifications occurs is rough endoplasmic reticulum, whereas the post-translational modifications occurs in rough endoplasmic reticulum, Golgi apparatus, endosomes, lysosomes, and secretory vesicles. And the process which are regulated by co-translation modifications are regulation of translation, protein folding, and processing, and some other. And the processes of post-translational modifications are phosphorylation, glycosylation, nitrosylation, and some others. Now let us enter into the post-translational modifications. There are mainly three types of post-translational modifications. That includes proteolytic degradation. And many proteins are synthesized as a precursor, which are much bigger in size than the functional proteins. Here, some portions of the precursor molecules are removed by proteolysis to liberate active proteins. This process is commonly called as trimming. The best example of this process is formation of insulin from pre-proinsulin and conversion of zymosomes, that is inactive digestive enzymes, for example trypsinogen, into active enzymes, or some of the best examples of proteolytic degradation. And the second one is intern splicing. Interns are nothing but the intervening sequences in certain proteins. These are similar to the introns in mRNAs. These interns have to be removed and the extends ligated in the appropriate order for the protein to become active. And the last one includes the covalent modifications, which we will be studying vastly. The proteins synthesized in translocation are subjected to many covalent changes. By these modifications in the amino acids, the proteins may be converted to active form or inactive form. Selected examples for covalent modifications are methylation, phosphorylation, glycosylation, ubiquitination, N-acylation, and proteolysis, etc. Here in this picture, we can see the examples for intern splicing and the proteolytic degradation. And here we can see that in intern splicing, the intern has been removed, and here in proteolytic degradation, when the signal sequence have been removed, the pre-proinsulin have been converted into proinsulin, and when the C-peptide have been removed, the proinsulin have been converted into active insulin. Different types of PTMs. There are many types of post-translational modifications. Here now we study about methylation, phosphorylation, glycosylation, ubiquitination, N-acylation, proteolysis, prenylation, hydroxylation, deamination. And the first one, methylation. Methylation is the best example for reversible post-translational modifications, and it is controlled by the enzyme methyltransferase, and it targets arginine and lysine. Methylation is one of the most common post-translational modifications in which the methyltransferase inserts one methyl group into amino acids. Methylation is an irreversible and reversible type. Here the SAM S-adenosyl 1-methionine get converted to S-adenyl homocysteine in presence of methyltransferase. The two common methyltransferases are PRMTS and HKMTS. Acronym stands for protein arginine methyltransferase and histine lysine methyltransferase. When the methyl group adds to the nitrogen, the process occurs irreversibly, but when the methyl group adds to the oxygen O-methylation, the process occurs reversibly. The best example to explain the methylation and demethylation of histones is histones of proteins are linked with the DNA and help DNA to package on chromosome via nucleosome assembly. The addition and removal of methyl group certainly affect gene expression. It regulates the number of transcripts formed during transcription. It helps in epigenetic regulations. Some of the advantages of protein methylation are: it helps in the regulation of gene expression, it helps in the protein stability, it helps in the processing of cellular processing, and it helps in protein-protein interactions, heterochromatin assembly, protein modification events, and epigenetic silences. Now we'll see about phosphorylation. It is also a kind of reversible post-translational modifications. Most studied post-translational modifications is of phosphorylation of proteins. It was reported by Phoebus Leveni in the year 1906. Two enzymes, it is first one is kinase and second one is phosphatase, catalyze the entire process of phosphorylation and dephosphorylation, etc. Adenosine triphosphate is the phosphate donor for the process to occur. Interestingly, it alters the protein function for a short time, meaning the protein function can be reverted when dephosphorylated. Protein phosphorylation is a kind of reversible post-translational modifications, adds and removes phosphate from the amino acids, especially threonine, tyrosine and serine. Phosphorylation is applicable in cell cycle regulation, growth and apoptosis, signal transduction pathways, activation and deactivation of enzymes, activation and deactivation of protein receptors, activation of cell membrane channels. And the next one is glycosylation. Glycosylation is similar to that of the phosphorylation. However, it is a bit complex. Glycosylation adds carbohydrate moiety to the amino acids of a particular protein, thereby forming an glycoprotein. Studies have shown that approximately 50% of our proteomes are various glycoproteins. It is a type of irreversible post-translational modifications. Enzymes involved in the whole processes are considered as glycosidases. Common to glycosylation types are N-linked and O-linked. Besides, the C-linked glycosylation and phosphoglycosylation and also glypation also occurs. The N-linked glycosylation occurs at the amide nitrogen of the N-terminal of amino acids, while the O-linked occurs at the oxygen atoms. The N-linked and the O-linked glycosylations occurs on serine and threonine respectively. The immunoglobulins, that is, antibodies, are the best examples of glycosylated proteins in which attachment of sugar moiety to protein forms different immunoglobulins. Glycosylation has also a major role in protein structure folding, conformation, stability, distribution and activity. And there are several applications of glycosylation which include: they play a major role in adaptive immunity, they help in the determining blood groups, and they provide structural support to membrane. The next one is ubiquitination. It is a type of reversible post-translational modifications. It was first reported by Gideon Goldstein in the year 1975. Ubiquitin is a protein that governs the catalytic reaction of ubiquitination that works in regulating and degrading proteins. It is an 8 kilodalton size small protein that binds to the substrate ubiquitin protein residue. The enzymes E1, E2 and E3 governs the whole process. Usually, the reaction occurs by forming the isopeptide bond between the ubiquitin and substrate protein at lysine, threonine, cysteine and serine residues. However, it may occur on all the 20 amino acids. The bond links the C terminal of the ubiquitin and the N terminal of the target protein. Researchers also show that a single or many ubiquitins may attach to a single target protein known as monoubiquitination and polyubiquitination. One of the major properties this reaction has it is the ubiquitinized substrate may further go into phosphorylation and acylation. The enzyme deubiquitinase receives ubiquitin from the target substrate. The process is mainly involved in the degradation of proteins, DNA repair signaling, inflammatory responses and organelle biogenesis. And there are some other important processes that includes regulation of transcription, replication and DNA repair and proliferation of signal transduction and apoptosis, and it helps in the major role of signaling innate immune system and intracellular trafficking and virus burden. And the next one is N-acylation. In 1964, Alfred VG has reported the first acylation modifications in the isolated Calf thymus nuclei. Two enzymes, CAT stands for lysine acetyltransferase and HAT histone acetyltransferase, have an important role in this process. The N-acylation process follows both reversible and irreversible mechanisms of post-translational modifications, transferring the acetyl group to the nitrogen residues of the target proteins. The process of chromosome relaxation and condensation is highly relying on acetylation. The lysine amino acids of the histone protein helps in chromosome packing, plays a vital role here. When N-acylation occurs at the lysine residue of the histone, it relaxes chromosome condensation and promotes transcription. On the other side, deacylation results in chromosome condensation and blocks transcription by dissolving DNA to transcription. HAT acetyltransferase and HDAC histone deacetylase co-repressor catalyze both reactions respectively. Importantly, the process known as hyperacylation maintains genomic integrity by a process called as gene silencing. The HAT uses acetyl-CoA as a cofactor to insert the acetyl group, while the HDAC removes the acetyl group from the lysine. The three common types of acylation are N-acylation, also called as alpha acylation, and N-epsilon acylation and O-acylation. This N-acylation post-translational modifications has a major role in the genome and chromatin stability, regulating transcription, regulation of cell cycles, nuclear transport, protein-protein interactions, and actin nucleation. The next one is proteolysis. Proteolysis is a process of protein degradation that occurs via the catalytic reactions of protease. It performs a vast array of biological functions. All eukaryotes have an enzyme protease present, which is largest group enzyme in humans. Post-translational modifications such as cleaving polyproteins, degrading precursors, signal sequence removal, removal of N-terminal methionine, and other protein degradation activates occurs by proteolysis. Here in the picture we can see that a protein by the action of protease is converted into small peptides. And the next one is prenylation. Prenylation is another type of lipidation. It occurs by the addition of lipids. It was reported by Uzuki et al. It's an irreversible post-translational modifications. 2% of all human proteins is prenylated. All proteins of RAS superfamily are mainly prenylated. The enzyme farnesyl transferase, in short called as FT, governs the catalytic reactions of adding the farnesyl or geranyl group to the cysteine residue of the protein. The prenylation post-translational modification has a significant role in various biological activities and protein regulations such as cell growth, endocytosis, protein-protein interactions, and trafficking of proteins. And the next one includes hydroxylation. The hydroxylation, the process of insertion of hydroxyl groups at the amino acid of a protein is called as hydroxylation. The enzyme hydroxylase governs the process that helps to convert a hydrophobic compound into hydrophilic. The best example of hydroxylation is collagen synthesis. It is one of the most common post-transcriptional modifications that adds functional group to the protein, thereby regulates protein-protein interactions, and it is an irreversible process. Deamination. The process of deamination is classified under amino acid modifications, which is conversion of amino acids from one to another form. For instance, asparagine to aspartic acid, glutamine to glutamic acid, or polyglutamic acids. And there is another type of modifications, and it is palmitolization. It is a kind of lipidation or a type of lipid modifications that uses palmitol transferases, in short called as PAT enzymes, for post-translational modifications. It was first reported by Bartels in the year 1999. Enzyme PAT adds palmital group of the palmital coenzyme A to the cysteine amino acids of the protein. It serves a permanent anchoring of palmitoylated protein to the membrane. The enzyme acyl protein thioesterases governs depalmitization process by removing the fatty acids from the protein. The process has functions in protein regulation, signal transduction, protein interactions, neuronal development, and apoptosis. The significance of post-translational modifications includes protein targeting, is also called as protein localization, protein signaling, that is protein-protein interactions and cascade amplifications, aids in proper protein folding. Few lectin molecules called calnexin binds to glycosylated proteins and assists in its foldings, confer stability to the proteins. Glycosylation can modify the stability of proteins by increasing protein half-life. It protects the protein against cleavage by proteolytic enzymes by blocking the cleavage site. Protein sorting or translocation. If phosphorylated mannose residues are present in the protein, it always goes to lysosome. It regulates protein activity and function. Acylation regulates many diverse functions, including DNA recognition, protein-protein interactions, and protein stability. Phosphorylation of protein is a reversible post-translational modification which activates the protein. Biochemical activity regulations, and redox dependent post-translation modifications of proteins is emerging as a key signaling system conserved through evolution, influences many aspects of cellular hemostasis. It significantly increases the diversity and complexity in the proteome. And the defects of post-translational modifications. Cancer and Angelman syndrome and lipofuscinosis are the major defects when the methylation process undergoes changes. according to the recent studies. The methylation errors are linked with several other types of genetic diseases. Error in the ubiquitination pathway causes inflammatory diseases, metabolic syndromes, type 2 diabetes, and cancer. A rapid aging, Huntington's disease, Parkinson's disease, immune disorders, and cardiovascular problems and cancers are some of the most common diseases associated with the dysregulation of acylation. Neurological problems and cancers are common diseases caused by the dysregulation of palmitylation. Abruption of prenylation may cause neurodegenerative, bone and metabolic and cardiovascular disorders. The summary: the post-translational modifications includes the covalent modifications as well as the intense splicing and proteolytic degradation. And in simple, we see some other types of covalent modifications which include sumolation. This process adds a small protein sumo. It is also called as small ubiquitin-like modifier to a target protein. And in hydroxylation attaches a hydroxyl group, and then methylation and methyl group is added, and the lipidation attaches a lipid. Acylation and aster group is added. Disulfide bonds covalently links the sulfur atoms of two different cysteine residues. And ubiquitination adds an ubiquitin to lysine. Glycosylation attaches a sugar, usually to the N or O in amino acid side chain, and phosphorylation adds a phosphate to the serine threonine or tyrosine. Thank you everyone. My special thanks to Dr. Murali Dhararao sir for giving me this opportunity to showcase my ability.